Okay, so this first part this morning is going to be non-technical from the brain simulation platform point of view. It is going to be closer to the experimentalists and to the neurobiology part of the school, in the sense that what I would like to try to do is to let you know what is the background to implement your own model of realistic model. When I say realistic, I mean something that you can compare directly with experimental data. Traces, features, behavior, and things like that. So let's start from the beginning. Suppose that you want to, suppose that you want to model a cell. It doesn't matter. I mean, in this case, it is a CA3 pyramidal cell from the hippocampus, but it doesn't matter. It could be amygdala, it could be basal ganglia, it could be neocortical, it could be any kind of neuron that you want. All neurons basically work following the same rules, okay? In the sense that they have channels, ion channels, transmembrane, that open, close, activate, deactivate, and also they alter the difference in the ion concentration between the inside and the outside of the cell. This generates some traces, some voltage that you can measure. In this particular case, what we had is a population of neuron in the CA3 region of the hippocampus that under the same current behave in a way that is completely different. Okay, so for example, we have a subpopulation that is weakly adapting. You have seen yesterday what means weakly adapting. In this case, it is even very, very weak. Delayed, adapting, or bursting. Okay, the proportion with which this is found in the slice is of course different. They are not 25%, 25%, 25%. They are going to be different. And but the real question here is that you cannot have. The morphology is more or less the same, but the electrophysiological properties of the channels that are expressed on the membrane of these different neurons must be different, because there is no other reason to explain why, under the same current injection, these neurons behave in a way that is completely different and independent from each other. I'm beginning to sweat now, so let's see. Anyway, so we wanted to study why these different neurons behave in different ways, so which means let's look at the channels that they are expressed in the membrane. It is known that they are expressed in the membrane, and let's see what are the composition and behavior of this channel put together in such a way that we can get the different behavior. So let's start. I mean, is there someone that has never done an experiment and a simulation neuron? So, one, two, so I need to explain almost everything very quickly, a couple of slides. What we do is take a reconstruction from, we pick a student, we put it under the microscope for a few hours and say, okay, let him do it, okay? And there are some problems with that, but at the end, you get a 3D reconstruction, okay? In which we have several different pieces of membrane split in points. Each point is modeled as X, Y, Z in diameter. You put everything together, including information on bifurcation, end, soma, axon, type of segment, and so on. And then we implement this in neuron as a piece of membrane which are small enough to be considered isopotential from a mathematical point of view. Okay, so for example, in this case, we have a cylinder that is 1.2 micron in diameter, 26 micron. This is our fake number, but in the real case could be very similar. This piece of membrane is assumed to be small enough that I don't see in the real neuron any difference if I record from here or if I record from here, which means that the entire piece is isopotential, at the same potential, okay? We put everything together using electrical equivalent of the circuit of the membrane and all the passive properties. And we also need to put active properties, because it is well known that these neurons, every neuron, at least in the soma, it has a set of ion channels. Ion channels are real holes in the membrane that let specific ions to go in and out of the membrane. You can see here in this case, in this particular case, these are, I think it is a tunneling microscope picture of an NMDA channel. And you can go to resolution up to the single channel. And you see that the difference between these two images is that this particular channel, in this case, is open. In this case, is closed. This is a computer reconstruction of the proteins that makes up the channel. They are proteins which are arranged in the membrane in such a way that they actually make a hole. Through that hole, ions can go in and out. When you change the difference in potential between the inside and the outside, what the proteins do is to change conformation in such a way that the hole is closed or open. It is a stochastic process. I will not go into the details, but Hodgkin and Huxley, I'm sure you know, in the 50s, arranged a set of equations that are able to replicate the macroscopic working of a set of channels in the membrane. Okay? Are you familiar with the Hodgkin and Huxley equations? I guess so, right? Except maybe for someone really not with a biological background, computer scientists and things like that. But I cannot go into the details. I can show you the equations. And the equation is something like this. On the top row, there is what is called the membrane equation. It is a way to calculate how the voltage, the difference in the voltage between the inside and the outside changes as a function of time according to different factors. Each factor is either an ion channel or passive properties or an input current. I mean, this equation could be as much as complicated as you want to be. Okay? Of course, as long as you have all the constants that define your equations. As long as you have an equation for any kind of biochemical signal that you want to implement in your neuron, you can put the equations as an additional terms into this equation, and you are going to add that mechanism into your neuron. Okay? A neuron does put everything together in a very nice and a very efficient way. So, for example, in the case of the sodium channels, you are familiar with this from Hodgkin and Huxley. These are gate variable. The gate variable derives from the probability of having the channel open and closed. You can express this in several different ways. These are not exactly the original Hodgkin and Huxley equations. This formulation is due to Borgram in the 70s, but it is very easy. What you need to get from the experimentalist are all these coefficients here. Once you get this, and you usually get this from the papers, and by the way, it has been about 20 years. The people, experimentalists, and I'm blaming the experimentalists here, don't want to do any more experimental work on ion channels. And the reason is that they cannot publish this kind of data on neuron, on Nature or Science, because they are kind of low level now for them. And they don't want to spend years following the difficulties in recording from a very small patch to get these coefficients that are fundamentally useful for the model, but they don't get to our impact factor journals. Okay? So these data are basically frozen up to about 20 years ago, at least for the hippocampus. I don't know, maybe in the basal ganglia or for the cerebellum, you have more recent data, or you do it in your lab anyway. So in particular, for the set of neurons that we were interested to study in this particular case, CA3 pyramidal neurons, there were no data. So what we did is, okay, let's start with the CA1 pyramidal cells and see what happens. Okay? Let's start with the sodium, KDR, and Ka. So there are three typical currents that are found anywhere in any neuron. The sodium is needed to get the action potential, and the KDR and Ka are needed to repolarize the neuron and get a train of action potential. Okay? So we will start from the CA1, but trying to model CA3 behavior. Okay, so let's start with the simulation. I'm going to arrange this to see. Okay, you can see both the. Let's see how it goes. Okay, let's try with the very first experimental trace. Okay, non-adapting or weak adapting. This is supposed to be the simplest one to model with only these 3 channels. And well, we can start right away with these numbers. You see here, these are the peak conductances. These are the kinetics. The kinetics are the inactivation, activation, and time constant for inactivation, time constant from inactivation, and the same for KDR and KA. If you don't know what I'm talking about, please stop me immediately, because things are going to get complicated. And if you are not following now, you are not going to be able to follow to follow me later in the talk. Okay, so only these 3 channels with these conductances. Let's see, and this current. Let's see what happens. Oh, it's firing too fast. Not too fast, but too early. The resting potential for this neuron is -60. The resting potential for the CA1 neuron is -65. So we are already close to the threshold. The CA1 neurons fire at around -54, 55. These neurons fires at -30. Okay? So it should be clear already from this very simple first simulation that there is no way that we can model the behavior of these cells using CA1 kinetics. Okay? Does anyone disagree with this? Good. So there is no way. So we need to figure out what's wrong, what we need to change, what is the minimum amount of change that we need to model at least this very simple trace. Okay? And by the way, let me see if I can do it without screwing up everything. Let me delete this. Okay. And then if I still insist and would like to change those numbers for the sodium, KDR, TDR, and KA in such a way to be as close as possible, at least with the firing frequency, the best I can get is this. Okay? With these numbers here for the peak conductance. So usually I can fit the first, the second, maybe the third, assuming some noise and things like that, but not much on top of that. And the major problem, of course, is the threshold. Okay, so the situation is so wrong that you should by now be convinced that I need to do something else. And this something else has to do with the threshold for firing. Okay? The threshold for firing here is -30. Suppose that I could play with the potassium conductance in such a way to hold the neuron until it reaches -30 to fire. Suppose. Even if I do that, I'm not going to get any action potential from the sodium, because if I slowly go to -30 for 50 milliseconds, like in the experiments, look at the sodium channel. At -50, at -30, the sodium channel is completely inactivated. You have no channels available to open and give you the action potential sweep up. Okay? So what is the solution for this? The solution is to assume, and again, there are no experimental data, so it is a prediction of the model. It is to assume that you need to shift all the channels by 25 millivolts in such a way that once you move all the kinetics to the right, you are going to have the activation and activation set up in such a way that they will open in the right range of voltages. Okay? Let me show you what happens if I shift by 24 millivolts. Okay, now you don't see it in the pictures above, of course, but I have a parameter here showing the 24 millivolts shift in that kinetics. In the next slide, we are going to see the shifted kinetics. But you see that now I can easily get the correct threshold for firing, and about, they are not right, but the frequency is, let's say, reasonable. Okay, it's not good, but it is reasonable. As a matter of fact, this is as far as I can go by manually modulating this peak conductance value. Okay, I could have been doing better, a little bit better, if I, in addition to change the peak conductance, I could begin to play with the kinetics. Distort a little bit the activation, move away the activation from inactivation, and play with the time constant and things like that. Okay, but in the absence of experimental data, the minimum amount of changes that I'm able to do and getting reasonable results, I think I will be happy with that. Okay, so let's go to the next slide and see where things are getting more complicated. Okay, forget about this second line and this Km here. Let's look again at the sodium KDR and Ka. Now everything has been shifted with respect to these plots here. Let's look at the KDR because it's easier to visualize. You see that the curve now, the activation starts at minus 30. In this case, is starting about -5, which is about 25 millivolts shift to the right. Okay, and in this case, so this is again the same thing as before. So now, what else I could change? I need to change. So the thing that you need to implement your model is a grasp of what each channel is going to do to the trace. If you are not able to visualize or predict what the current is going to do to the trace, then you better go to the blue pie optimization and let him do all the work. Okay, this is what Carmen is going to show you later, but let's stay on the neurobiological point of view, and let's try to figure out what each current does to the trace in such a way that we can think in advance what we need to change to get as close as possible to the experiments. So let's come here, and okay, let's do some experiments. Let's start from this trace here. Okay, so suppose that I change the KDR or the Ka. What do you expect? Anybody would like to try? Don't hide behind the other people in front of you. If nobody would like to try, then let's try it on the simulation. I'm just going to say, okay, I change the KDR.
Change the frequency of
It changed the frequency. It changed the interspike interval. Any fast potassium current. Yes, you want to say something? Yeah. Ignore the Km and the other currents on the bottom row. We are going to get there in a few minutes. Let's talk about sodium KDR and Ka. Sodium is sodium current. KDR and Ka are two different potassium currents. There are plenty of potassium currents in the channels in the neuron. Okay? In this particular case, the KDR is the one that is also being studied by Ogden and Huxley for the squid axon. It's a non-inactivating, which means that if you depolarize the cell, this channel is going to be open, and it stays open until the voltage is there. Okay? The Ka instead is fast, which is faster. You see that this is twice faster than the KDR. It also has an activation and inactivation, which means that if you depolarize the cell, the current first opens and then closes. The KDR instead stays open. So you have a constant flux of potassium ions when the KDR is open. It is a transient current for the Ka. Okay? Okay, so if we increase, as your colleague was suggesting, if we increase either the KDR or the Ka, what you expect, based on the kinetics, is an increase in the interspike interval. Okay, so let's try that. And in fact, you say now, I mean, it is starting late, and there is going to be another spike around here. Not because I'm a magician, I tried before, okay? So I know what is going to happen in most cases. And so you see, let's do it. Okay, you see, interspike interval is changed. This depends, of course, on the conductance. Let's do the same for the KDR, for the Ka. This is more tricky because this is a stronger current, so we need to go a little bit. So again, in this case, there are going to be a couple of spikes with a larger interspike interval. So these two currents are going to change the interspike interval, the frequency at which maybe we should open the door so we can allow some air coming in and out. Otherwise, I know, but I'm melting anyway. Yeah. Okay, thank you. In CA1 neurons, the Ka does much more, but for other reasons. I'm not going to explain you in details what the current distribution is in the CA1, but the CA1 neurons, pyramidal neurons in the hippocampus are the most complex from this point of view because they have a nonlinear distribution of channels in the dendrites that makes a lot of computational power, gives a lot of computational power to these neurons, but I'm not going to go there. Just to say one thing, the fact that the Ka is fast and transient allows you to modulate the amplitude, the duration of the action potential, okay? And the duration of action potential means a different calcium entry when you are going to go and study plasticity and things like that. But we are not going to do here now. Let's see what happens in the adapting. No, let's forget about the delay. The adapting, okay? So now you see that the interspike interval is not constant anymore. It is increasing with time, okay? And since it is increasing with time, it means that during the simulation, something is changing. Something is not going back to the original state, okay? And this cannot be done with these currents. Because with this kind of time constant here, 14 milliseconds, 30 milliseconds, down to a fraction of 2 milliseconds or 1 millisecond here, they are too fast. Within the repolarization phase of the action potential, all the current will basically shut it down, okay? Under this condition, after each spike, the conditions of the voltages are going to be back to the resting voltage or to the equilibrium voltage. And so each action potential is going to be at exactly the same frequency as we saw before with the non-adapting case. So we need something else. This something else is caused by another current. Let me close this for a moment. Let's look at the Km. Yet another potassium current, okay? In this case, it is also non-inactivating, but look at the time constant. The time constant now at physiological temperature is order of 100 to 200 milliseconds. So it is much, much slower current, okay? And it is lower overall the entire range of voltages, which means that suppose that I go from minus 65 or minus 60, which is the resting potential, I will get an action potential, so I go to 30 millivolts, okay? And I go here in a matter of milliseconds. So the current does not have the time to go and open completely because it is too slow to follow the voltage. However, it goes some way, okay? So let's say that it opens a little bit. From its state at minus 60, it tries to open a little bit and stays here. And when the action potential closes, it does not have the time to go to the resting potential again, to close again, because the time constant is about 50 milliseconds. Okay? And so at each action potential, this current is going to be open, more open, more open, more open, more open, in such a way to have a kind of memory. It's called hysteresis in physics. It goes one way, but it does not go exactly the same, along the same pathway backward. And so what you get is a potassium current that at each action potential is going to be open, more open, more open, more open, and you get the increase in the interspike interval. Okay? Do they decay? Yes. If the current is small enough, then you let the current to go back to its resting state, and so you are going to have a periodic action potential and the constant current that are not going to show any adaptation. Yeah, but I mean, it is always a dynamic feature. So if you have a current which is strong enough to open the to open the Km, it is going to increase until it reaches a steady state. So what we get again with the simulation, if we put also the Km, it is something like this. So adapting and we get something like this. And now we have only the Km current. With only Km, you get this spike, then a spike which is laterally delayed, and then big delay. Of course, if I reduce here the potassium, the Km, I would get also a different effect. Oh. I reduced it from whatever it was to 0.6. Now I get less adaptation. You remember that there was a third and a fourth spike that was way up. In this case, we should get more spikes. Yeah. Okay, because I've reduced the effect of the Km by reducing the peak conductance. And so the best I can do with only Km is the yellow line that you saw before. Let me show it again. This is the best I can get by tweaking a little bit the numbers. It is not good enough. Okay? Even if I'm going to show you later that it is useless, even if I want, let's say that I want to model exactly these trays, okay? It is useless. It is not worth it. But still, I want to do it, okay? Because I know that some experimentalists say, okay, this is my trace. It's the Bible. I want to, or whatever religious stuff you have, and I want exactly these trays to model exactly my neuron. Okay? There is no way I can do it with the Km alone. I need another mechanism. In this case, another mechanism that can add another piece of memory, short-term memory, to modulate the interspike interval. And this is a calcium-dependent, again, potassium channel. Okay? This KCA has a time constant about 80 milliseconds, so it is large, but this is not the main reason to use it for increasing or modulating the adaptation. The main reason is that the activation kinetic shifts with calcium entry. So now the memory mechanism is not anymore the timing and the progressive opening of the current with time, but now the current is going to be open with calcium. The more calcium you have in the cell, the more it shifts, and then at the same potential, you get an activation that is going to up or down following your calcium concentration. Okay? And in the model, there are a few calcium channels, They are basically the three main types of calcium channels that are in the neurons, which is a long-lasting current, a transient current, and neither or two. Okay, you can model the entire set of calcium behavior by adding to your model these three types of calcium channels. So let's see how far we can go with this. Okay, adaptation is this, and now I'm going to use only the calcium-dependent potassium channel, and I get this. Again, alone, I cannot get my trace. However, if I use both, and by both mean both the Km and the calcium, now I am satisfied, okay? Because by arranging the peak conductances, I can reach a point in which I can model more or less exactly my trace. In order to do that, however, we need to be clear. I just follow a logic path that tells me that each current has its own role. I'm not using more, I'm not using less, okay? This does not mean that the neuron has exactly those currents and exactly those conductances, because you know that if you do patch recordings from different neurons from the same population, you are going to get conductances that are not identical. They are quite different. Even if you look at the sodium channel, you are going to get a distribution of values. If you get if you measure the KDR or the Km or the potassium-dependent calcium conductance in each neuron, you are going to get a bunch of different values that could be quite different from each other. Okay? So even if I model perfectly my trace, this is going to be for this trace. It does not ensure me that with the same set of conductances, I'm going to get perfect match with another trace from another neuron, even if it is from the same population. Okay? And so we can go on, of course, and if I now want to model the bursting behavior, which is a very extreme and borderline behavior, which means that anything I change a little bit there, the trace is going to undergo a quite substantial change in shape. Then I can, again, use all this bunch of current, but I can reorganize the conductances in such a way to have a good replication of my data using a different set of numbers. But using always sodium, Ka, KDR, Km, and Kca. Okay? So we were satisfied with this because now we had a way to model the different behavior of different subpopulation of neurons and getting the different traces. Bursting, not bursting, adapting, delayed. I didn't show the delay, but the delay traces are simply caused by a single current called Kd for delay, that it is known to be in these neurons, and causes a delay in the first spike. Okay, so what we did now is, okay, let's see not only the single voltage, but how they are organized, how they are connected to each other, if there is any connection. So we did a simulation in which we took all the all the conductances, a bunch of current injection, and we, of course, we dumped everything on the supercomputer and did about 20 million combination of conductances, currents, and classifying the output based on the spiking times in terms of bursting, not bursting, delayed, adapting, not adapting, and things like that. And we analyzed the results. So this is a loop showing all the possible combination, the kind of traces that you get. Some of them are ugly, of course, but don't tell me that in the experiments you don't get ugly traces sometimes. You do, right? And but these are taken care by the model. Those are caused by strange arrangement of conductances. And so once we get all these traces, of course, most of them were nonsense, about below 100,000 were classified by the algorithm. And we can analyze the results and figure out how they are organized. And so, for example, let's take the delayed traces. In 40% of them, of the cases, we can model delayed traces with only the KD. It doesn't matter what they are, what the other currents are. In an additional 25-30%, we need the KCA. In other percent, we need the KM, and so on. There are no traces that could be modeled, no delayed traces, that could be modeled without the KD. So the KD is one specific current that is needed if you want to delay, of course. Okay? Adapting. The adapting, weakly adapting or strongly adapting, requires the calcium-dependent potassium current in 95% of the cases. Okay? So you can model 95% of the cases with a calcium-dependent potassium current which is larger than Km. You cannot model, essentially, these kind of traces if you have a conductance or calcium-dependent conductance that is lower than Km. So this is another prediction for the experimentalists. If you go there and look at the patch clamp in the sodium, in the soma of these neurons, our model is predicting that for the adapting neurons, you should have always, almost always, the calcium-dependent potassium current lower in conductance with respect to the Km. Okay? And so on. For the bursting is the opposite. So adapting is calcium-dependent greater than Km. For the bursting is the opposite. Okay? So let's say, okay, I modeled one cell, but I want to model an entire population. And so if you do the experiments in several neurons, you see that there are a distribution of channels, large distribution in the values. Why they are so distributed? Why they are not identical? Okay? In the face of morphologies that are similar, also slightly different. So the question here is that during its lifetime, a neuron needs to adapt its input-output properties because it is part of the network. And if it is part of the network, it means that it must be something with the input and give a specific output. If it deviates for some reason, pathological or some other problems, it does not give again the same output, there is going to be a problem in the network. Maybe not for a single neuron, but when all the neurons are going to be altered in the normal functioning, for example, in Alzheimer or in some other pathologies, the network is going to not work good enough. And so there must be some mechanism that keep the neuron working within that I/O properties by compensating for some change in one piece or one part. So for example, if we have, and there are some pathologies like, for example, typically epilepsy, which is known to be dependent on, at least in some cases, what is called febrile neonatal epilepsy. The medical term, it is something like this. It is known that is caused by a mutation in the Km channels. Okay? They are changing the kinetics. In order to have the neuron to work, I mean, you know that the brain is a very robust entity. Okay? If you take out one single neuron, it doesn't happen much. If you take out, if you take a Google car and take one transistor, you don't know what's going to happen. Okay? Most likely, it will stop forever. Okay? In the neuron, you can do this very easily because there must be some mechanism that is responsible for taking care of the different configuration of the neuron under pathological or physiological condition. Okay? So basically, what the neuron does, what nature does, is to have all these conductances to compensate each other in such a way that the neuron, no matter how strong you take it as smash on the floor, is still or better, you cut the brain, you put in the eyes, you cut a slice, you put a solution, you put under the microscope, and it still is living for a few hours. There must be some strong compensatory mechanisms. Okay, so we decided to study this effect. That, by the way, in physics or in system biology is called degeneracy. Degeneracy, which is a phenomenon allowing the possibility for a complex biological system to perform the same function under different structurally different elements. In this case, our elements are the ion channels. And so we wanted to study how many possible combinations of these channels allow us to have the neuron to work to be consistent with the traces that we see in the experiments. Okay, so we took a bunch of morphologies. In this case, 34 morphologies, 19 pyramidal cells, and 15 neurons from the CA1 regions of the hippocampus. Does everybody knows what the hippocampus is? Who doesn't know? Okay, good. So we took these morphologies, and for each of them, we try to implement, these are experimental traces. We have about 1,500 experimental traces that were used to extract about 100 electrophysiological features using the software that you saw yesterday on the platform. And with those traces and those electrophysiological features, we were able, and with the BluePy-OPT, the BluePy-OPT is an automatic optimization program that Carmen is going to show you not only this morning, but also with hands-on this afternoon. It is possible to, let's say, guide the optimization process by telling the program, okay, on these neurons, I want to have these kind of channels with this range of conductance distributed in this way. Do it whatever you want, but tell me in how many possible ways you are able to set up these conductance, these numbers, these variables in such a way to be consistent with all the electrophysiological features that we selected from those 1,500 traces. Okay, and the optimization works with following a genetic algorithm. So you get several different individuals which are good enough, and from this good enough individual, we can analyze the results. Okay, and in some cases, we can also predict some of the behavior in CA1 neurons that were not imposed on the optimization side, and we get this. This is quite complicated, but let me spend a few minutes to explain you what this figure is. First of all, let's concentrate on pyramidal CAC, pyramidal cells. Okay, so what you see here in the x-axis are the different individuals that fit more or less equally well all the traces with all the electrophysiological features. Okay, and we have about 160 different configuration of peak conductances of all the channels that we decide to put in the CA1 pyramidal cells. And the name of the conductance is here in the axis. We use two axes, but they're the same, just because all of them cannot be placed on the same axis. They are normalized values. So black means low value of conductance, low with respect to the range that we have set up, and red means high. And so, for example, if you look at the single optimization, all these colors here means that the Km in the soma has a low value, the Km in the axon has a low value, the EPAS, is the reversal potential of the resting potential, basically, of the leak current, is high value, the H current has a high value, and all the other are mixed. Okay? Instead, if you look at the horizontal line, this will tell you that those specific currents don't change too much from optimization to optimization. They need to stay at the high value. the Km, for example, must stay at a low value. All the others can do whatever they want. Okay? What this means? This means that there are some properties, some active properties of the neuron that must be constant. They cannot be changed. Otherwise, the neuron is not consistent anymore with experimental data. In this case, is the Km, either in the axon or in the soma, the IH, or the passive properties. Okay, these are either consistently low or consistently high. So in any kind of combination that you may want to use to fit your model with experimental data, you may have a very large distribution of values for a few conductance, but there are a few that must stay there, cannot be changed too much. It is surprising for us, it has been surprising, that these currents for which you don't have, you must have a constant value, are the IH and the Km, which both are known to be responsible for epilepsy. Okay? So this is a reason that explains why a single mutation in one of these channels, IH is one, but also the Km, will move the IO properties of your neuron, CA1 pyramidal neuron, from physiological to epilepsy. Okay? All the others can be adjusted. All the others, the neuron can say, okay, there is, for some reason, especially because of protein turnover, protein phosphorylation activity, all these biochemical processes are going to change the density of the channels in the membrane, independently, no matter what. The very simple fact that the neuron is alive means that it is going to change, like we change. I mean, hair grows, fat instead of not fat. But I mean, the neuron has the same problem. Okay, it needs to adapt its properties. It can adapt anything except a few conductances. Those must stay the same. Otherwise, you will get in trouble. And the same for the interneurons. Okay, so back now to the platform. I think that Luca yesterday showed you the results, the live collab for this paper. And this is because, as usual for the Human Brain Project and the brain simulation platform in particular, every time we have a paper, we have a EU-funded project. So we are obliged to basically make it available to everybody. In particular, for my model, all the models have been on ModelDB since 20 years ago. But in this particular case, we have set up the brain simulation platform with a link called the live paper, in which you can look at all the data that are in those used for this paper in particular. Okay, I'm going directly to the link, but if you follow the link from the brain simulation platform, you see that there are live papers, and then this is the first one so far, and so it goes to a collab in which we have placed everything, which means not only the abstract, of course, and the bibliographic information, the links and everything, but also all the data that we use. So, for example, we use all these morphologies files. So you can download it or view it directly using the one of the tools that are already in the platform. So this is the neuron here, and you can rotate, zooming, and eventually look at it and say, okay, I like it, I can download it and play with it, or I don't like it, go out and check out another one. The same with the traces still not visualize. We cannot visualize the traces now, but we are working to this purpose using the feature extraction program or some part of it to show the traces and eventually to download. But you can download the ABF files. So these are the raw data. I don't know what you are going to do with it because this is a proprietary. Don't tell them. Usually the experimentalists don't want to share the data. Okay? As a modeler, I don't care. But if you ask the experimentalist, could you give me your data? I want to use it for the next 100 years. I can get a lot of papers out of them. Probably they don't, but still there are problems. Within the Human Brain Project, this is not to be the case. Every time we publish a paper, everything is public, okay, including the experimental data. And of course also the simulation. You can go to the model catalog for all the optimizations, or you can go to ModelDB to extract some of the configuration. But a few simulation, you can run it from here using the Neuron as a Service. Did you show the Neuron as a Service yesterday? Okay, from this
Yes.
collab. So you already saw that. This is the same software that you are going to see in the tomorrow in talking about the small circuit in silico experiments, and also for the single cell experiments by Jean Denis. I don't know, Armando. No, Armando is going to talk about the circuit itself. Okay. Okay, so let me close this. Let's go back here. So what is the next step now? The next step, after you have your model, you cannot stop there. Yes, okay, you publish a paper, but then you want to do something, okay? In our case, we have a bunch of optimizations, so we have several different morphologies for the same area, interneurons, pyramidal cells. We have several morphologies. We can clone the morphologies, and we can build the entire CO1 region. This is going to be shown tomorrow by Armando, right? You're going to do the circuit tomorrow. Yeah. So the entire process that we call the workflow to start on the single cell optimization, that build the entire circuit, is going to be detailed tomorrow, and in the afternoon tomorrow you're going to see how this could be run in a supercomputer. But in our case, this is, I can skip this. By the way, you that are not Italian, you know the hippocampus word, right? The hippocampus is Latin for the seahorse. That's why it's called hippocampus, because Carl first looked at the hippocampus and said, okay, looks like this structure. And he said, this looks like a seahorse, but the Latin word for seahorse is hippocampus. That's the reason why it's called hippocampus. And you see here why. So the CO1 is on top of this, on the external surface of this structure here. And this is the current version of the model. It has about 450,000 neurons between pyramidal cells and interneurons. Including the synapses, now we are about 2 billion equations, ordinary differential equation, non-linear. Okay? So it is not something that you can run on your laptop. You need a supercomputer. That's why, and you're going to play with it a little bit, at least for a subset of cells, using some of the tools that are in the brain simulation platform. Of course, you are not going to run the entire circuit, because it takes 1 second of simulation time, takes 5 hours on 7,000 cores. Okay, we don't want you to use that much time of our time. So, but we are going to have some time to use and do something. What we want to, oh no, I can show you the movie of the entire beast. Okay, so this is growing. I mean, just adding cells until it reaches the 400,000 cells, and then I will go back to a little and a smaller number of cells in such a way to show you the activity. So this is what happens. You see those spikes there? This is what happens as soon as we put the system together. And of course, it has been validated against experimental data in terms of connectivity, background activity, and things like that. In this case, those spikes that you see in those neurons are because the minis activity. Okay? So there is a background with no external input. But as soon as we put together, there is an emerging properties that you're going to see here. Yeah. So the system becomes alive, basically. There are traveling waves, which has been observed in the hippocampus. And if you get the frequency, that you do the power spectra of this, you figure out and you see that they are a theta rhythm, just like seen experimentally. And this is without any external inputs. Okay? So one might say, okay, yes, this is because there are some hotspots But what if you, for example, activate a slice? You see a simulation here?
Are those resting state activated?
Excuse me?
Are those resting state activated?
They are resting states. I mean, there are no external inputs. There are only intrinsic synapses.
They are learning.
They are not learning. There is no plasticity here. This is just background activity that generates some spontaneous activity that travels along the hippocampus. Yeah.
Are there some like pacemakers or?
No, there are no pacemakers. We are going to figure out and publish a paper as soon as we figure out why. And but this is exactly the same as observed experimentally. There is a paper showing 2011, right, Armando? The Gutagni paper is 2009. They took a hippocampus, they cut and isolated the entire hippocampus. They cut and isolated the entire only the CA1 region, and they saw traveling waves at theta rhythm. Okay?
But some of the cells are just active.
Yes. But once you cross the threshold, the theta waves emerge spontaneously. Now we are going to study why. What is the underlying mechanism? But because there are no pacemaking cells, but there are hotspots. You see, for example, this and this are preferentially where the wave starts, also in the middle. And so even if we cut the extremities from there, you are going to see in a moment that you still get some spontaneous waves. You see, we cut everything. You don't get the traveling waves from the right and left, but you still get something there. And so we are going to study what are the mechanisms, the balance in between excitation and inhibition, which neuron population is most responsible for the origin of this kind of stuff, and how we can get to cognitive processes. Okay, spatial navigation, Alzheimer, epilepsy, and things like that. As a matter of fact, what we want to do at the end is this. Okay, so we have a model which is large, of course. We need to reduce it until we can put it in a chip. This is something that Egidio is doing for the cerebellum, for example. And once you have this, your reduced model, integrate and fire, or a single point neuron, or whatever you want, it is not something that you built on purpose. It is something that comes from the biology. And so I think that, we claim that it is a better model because it gives you the real algorithm used by the system. It is not going to be perfect for years. If you, for example, there are deep learning algorithms that work beautifully. You take, again, the Google car, okay? It can go around in the towns, but take the Google car and bring it to your backyard. What do you think is going to happen? It's going to bump around all the walls, okay? And this is because the Google car does not use the navigation that we use for the hippocampus. If you go here, you've never been here, but you were able to navigate the hotel without any problem because you have the hippocampus. You don't have a deep learning algorithm. This is the reason why we want to do this rather than deep learning and artificial intelligence like anybody else is doing. Okay, I think I'm done with 20 minutes ahead, but I expect a lot of questions. The hippocampus, of course, it's not something done only in my lab, but there is a big group of people in different laboratories. Of course, the Blue Brain Project at PFL, and the UCL in London, the Institute of Experimental Medicine in Budapest, and my lab. Of course, from EPFL, there are a bunch of people that are working very hard to build all the tools that you see, and many more, that are needed to build the circuit automatically from the data. We call it data-driven modeling for this reason. They are doing the cortex. Another group is doing the basal ganglia. Another group is doing the cerebellum, and we are doing the hippocampus. And finally, thank you to all for my lab. Most of them are, of course, here, except the two late entries. I did not have the time to include you in the picture. And thank you very much for your attention.